He kōna ipurangi tēnei nā te reo irirangi o Aotearoa.
With Ectotherm keeper Chye-Mei Huang, I'm climbing into an enclosure in Auckland Zoo to meet some of the special reptiles they have on display.
Everybody's hungry.
Oh, so you've got a little pottle with you.
Yes.
And what have you got?
Just a few little locusts here, some very, very tiny ones at the moment. He's used to much bigger ones, but he's also very food motivated, so he'll probably chase these, and I need him to get as much exercise and enrichment out of his feeding as possible. So lots of little ones today.
I can just about see the edge of his nose, just poking out there. Oh, he's interested.
He's like, those are tiny.
Why, do you think he's turning up his nose? He's used to the bigger ones.
Yeah, he's wondering if it's worth going for a run with something so little.
Kia ora, nau mai haramai ki te ao hurihanga. Hello and welcome to Our Changing World, ko Clegg and Cannon tēnei. This chilled out reptile, who isn't all that bothered about these tiny locust treats, is one of the 2 tuatara on display at Auckland Zoo. Tuatara are only found in Aotearoa, New Zealand, and they are the only living species of an ancient order of reptile. Their ancestors lived alongside dinosaurs, as far back as an estimated 240 million years ago. All of the species in the order died out 65 million years ago, except for the proto-tuatara, and it seems they've changed very little since this time. That's why they're often referred to as living fossils, and why some biologists find them particularly fascinating.
Tuatara are really evolutionarily interesting.
That's Cam Hoffbeck, a PhD candidate at Waipapa Taumatarau, the University of Auckland. And she's pretty familiar with these 2 specific tuatara, one male, one female, because they've been a part of her research over the last few years, which we're about to get into, but first, let's properly meet the tuatara. Their home is in the Islands area of Auckland Zoo. There's a space set up like an old Department of Conservation field research hut. A green and yellow sign on the outside, wooden framing and canvas tacked to the roof inside. Both walls are lined with glass displays looking into compartments filled with sticks, rocks, leaves and plants. And in front are placards telling you who's inside if you look closely. On the left-hand side, a gold-striped gecko, phalla skink and an elegant gecko. And on the right, one big enclosure lined with dried leaves and dead branches with a little stream running through. And it's this enclosure that Chye-Mei and I have climbed into through a hidden door at the back that she uses to do the feeding, cleaning and monitoring. What's the day in the life for a tuatara in Auckland Zoo?
They are very energy efficient, so a lot of people often ask us if they're real, because they do spend a lot of time sort of sitting around and resting. If they don't need to do anything, they won't, which I think is quite sensible of them. But equally, they're all about environment, so their environmental needs. So most of the time when you see them moving around, they'll be heading towards, say, a basking spot, which we've provided in here, or like this fellow here, he's covered in mud, and he's been in his burrow, so I can tell that because it's a little bit warmer today, he's probably been doing a little bit of digging and some burrow expansion to get down to the nice sort of cooler areas that he can hang out in when it gets really warm come summer.
Right, because this is, when you say ectotherms, this is the thing we would have called them cold-blooded before. They've got to regulate their own temperature.
Absolutely, and they can do that basically by finding the areas in the environment that best suit what temperature they want to be at and moving there, and then they'll take on that environmental temperature.
The male tuatara has shuffled out a bit from his hiding spot, greenish-brown with spines along the length of his back. They look intimidating, but they're actually quite soft spines, says Chye-Mei. There's a group of very excited kids in the hut area now, looking in through the glass and pointing.
Is that all for today? Fair enough. So it's all about choice in here, really, just providing him with anything that he might want to do or interact with in the course of his day. We don't make him do anything he doesn't want to, and he sort of just chooses whatever he gets up to. Today that's sitting at the entrance of his burrow, covered in dirt, judging us a little bit, probably.
Observing the miniature locust
Yes.
with disdain.
Yes. summertime, that will definitely change. He'll be nice and warm, and he will be keen as beans for pretty much anything he can fit into his mouth.
The female is keeping to herself in her own burrow. Done for now, we head back out through the small back room, and just as we leave, I capture this perfectly normal snippet of conversation between Cam and Chye-Mei.
How have you been? I'm afraid I've only got one frozen poo for you.
That's completely fine. I
At the
appreciate
moment,
even the one
things are starting to ramp up in terms of poo production.
Yes, the more poo you can give me, the happier I will be.
Sorry, this is tuatara poo. Yes. Should have maybe clarified that.
Our biologists. But it does have reference for Cam's work, because she's not actually focused on the tuatara themselves, more the community of living things that they are a home for. We sit down on a wall outside to chat.
The gut microbiome is all the little bacteria and other little tiny organisms that are living inside your digestive tract. And we all have a gut microbiome, but there's a lot of interest in how that might have evolved through time. If you think about like bees and flowers, the honeybees have those little fuzzes on their backs, specifically to collect pollen from the flower to pollinate, and then they have a tongue specifically to get into the flower. So that's a symbiosis that's evolved through time. So without flowers, there would be no bees as we know them today, and vice versa. But then when we're talking about the gut microbiome, that's also a really tight symbiosis, but it's between potentially one organism on one side and then thousands of organisms on the other side. So it's a much more complex situation. So for something like tuatara, which is very evolutionarily distinct, that is a very interesting thing to study for how its gut microbiome looks and what it might tell us about a potentially ancient gut community.
Because you might have microbes that are only found in the tuatara? Is that why it's particularly interesting?
Yes, potentially. And then tuatara also have a very interesting ecology. They're super long-lived, so we know they can live to over 100 years in captivity. We don't really know their lifespan in the wild. And they're also a cold-adapted reptile, which is really rare. It's not that rare here in Aotearoa, but in the rest of the world it's pretty rare. So they're very interesting, and they've also been in Aotearoa for so long. They're clearly successful here, so anything that could give us more clues as to why they live the way they do and their ecology and their specific evolution is really interesting from a bacterial point of view as well.
So how do you go studying the gut microbiome of a tuatara?
It's a great question that I also had to ponder at the beginning of this PhD. So kind of the gold standard in the field is fecal samples for gut microbiology, because that gives us a snapshot of what bacteria were living in the higher gut. So if you can't take an actual gut sample, like a tissue sample, which we would never do in case of a tonga species like this, you would want a fecal sample, but tuatara as reptiles just poop very rarely. So I can't reliably get fecal samples.
What's very rarely?
So like Chye-Mei, who we were hearing from, she's been on the lookout for a fecal sample for me for months. So they do poop, but they often do it in water or just so rarely, or by the time anyone notices it's too old and I can't use it anymore. So it's actually shockingly difficult versus if you're working with birds or something where when you pick them up, they tend to just deposit a sample right on you in the moment. So I have a much more difficult proposition there.
Right, and that's why she was saying that she had poo for you in the freezer.
Yes, yeah.
Which is a source of excitement.
A great excitement for me, yes. And then a lot of my work is in the wild, where there's even less chance that in the 4 minutes that I'm handling an individual tuatara, it's going to poop while I'm able to get a sample. So for me, I use cloacal swabbing. So the cloaca is the shared excretory and reproductive opening on reptiles. And then I just basically take a COVID swab of the cloaca. So just real quick, it's minimally invasive, so it doesn't harm them. And then I just get a little proxy of what bacteria might be living in their lower intestinal tract.
Today, tuatara are found in the wild only on offshore islands. Rats and mice prey on them, so you only see them on the mainland in predator-free fenced ecosanctuaries or in display enclosures like this. For her project, part of what Cam wants to understand is whether the shift to ecosanctuary or zoo life is going okay microbiome-wise for the tuatara. So she's looking at samples from a number of them.
I have colleagues who work on Takaporewa or Stevens Island, which is the source population of most mainland tuatara today. So back in the day, there would have been tuatara all over New Zealand, but they went basically extinct when humans started settling here, especially when European settlers established, they would have been extinct on the mainland. So all modern tuatara are descended from ones that survived on a handful of offshore islands couple hundred years ago. So I've studied those tuatara, but I haven't gone there myself because I don't have helicopter money in my grant. But I have colleagues who look at those ones for me. And then my first study was looking at the tuatara at the 5 different mainland fenced-in sanctuaries. So that's Sanctuary Mountain Maungatautari near Hamilton, and then Young Nick's Head near Gisborne, Cape Sanctuary near Napier, Zealandia in Wellington, and Orokonui in Dunedin. So that was my first study. And then in addition to that, I go back to Zealandia seasonally. And then I also did a study on the diet of tuatara at Auckland Zoo and Wellington Zoo and Victoria University of Wellington.
So first, looking at the cloacal swabs that come from the wild tuatara versus those ones in captivity, is the thought here that the microbiome could be different? I mean, how quickly does a microbiome change? You know, how long have these tuatara been in captivity, and would there be a big enough change within that time?
Yeah, so the tuatara that are here have been here for quite a while. I don't know the exact number of years, but they've definitely been here for multiple years now. And the gut microbiome can shift pretty quickly. So in humans, I became vegetarian semi-recently, we'd expect the gut to shift within a couple days to a week of an intervention like that. So the gut is pretty dynamic and able to shift to whatever, because you can imagine what you're eating is directly feeding what's in your gut. So if you stop feeding it, it's just going to pass on through. So with animals in captivity, we do expect to see a fairly different gut community. And that's a factor of the environment that they're interacting with. They're interacting with different or fewer species in there. They're interacting much more with humans than they would be in the wild. So sometimes we see a shift more towards human-associated bacteria. They're also eating different invertebrates. They're interacting with different plants, things of that nature. So completely different environment and diet. Even if you're doing the best you can to simulate the wild environment for their enrichment, you're still potentially not bringing in those bacteria. And often that's not even desirable for zookeepers. They don't want to be bringing in soil from the wild that might be contaminated with pathogens or something like that.
And so did you find that? Did you see a difference in the microbiome between captive and wild tuatara?
Somewhat. So it was a, p less than 0.05 situation.
A p-value is a statistical value that you get after you've run analysis to tell you whether something is different or not. And a p-value of 0.05 or lower is generally considered statistically significant. So what she's saying here is that she did see a statistically significant difference.
But it wasn't a very big effect size. And then the differences that I did see were in what we call relative abundance. So the amounts of the different bacteria that were present were different, but there was only one new genus of bacteria in the captive ones versus the wild ones. So overall, they have kind of strikingly similar gut communities, much more so than I was expecting.
Did any kind of new or unusual or never seen before things pop up?
Yes, and maybe. Yeah, so my first round of sequencing I did with Illumina sequencing, which is sequencing a little tiny fragment of the 16S gene of bacteria. So it's a gene that basically every bacterial species has, so I can be confident that that gene is going to be in what I'm trying to sequence. But it's so short that it's very difficult to get species-level assignments out of that, so it'll just tell me kind of generally what the bacteria was. But when I did that sequencing, it told me that it couldn't tell what genus most of my bacteria were. So that's pretty exciting for a first-year PhD student to realize I had a bunch of potentially undiscovered bacteria on my hands. But then I've since started sequencing longer fragments of the gene, still just the 16S gene. So with that, I've been able to get more resolution and figure out what I suspect some of those are. But even the ones that I do know what they are very different from, especially like a mammalian gut, but it's also very different from the guts of other reptiles that I've investigated.
Right, because I mean that's the interesting comparison. You would presume that it would be different to a human or
yes,
mammalian
certainly,
gut.
yeah.
We're doing quite different things, but yeah, so it's even different to its reptile. I mean, I was going to say cousins, but like you've explained, evolutionary-wise, they're quite different.
They are, yeah. So their closest living relatives would be the squamates. So that's things like skinks and geckos and snakes. But yeah, it didn't even look remotely similar to those. There's basically a completely different set of dominant bacteria occurring in the tuatara gut.
And now you'll run up against the problem where we don't know what we don't know, right? Because when you sequence the bacteria, you're comparing it to existing libraries of bacteria that have been sequenced before. Mm-hmm. Whereas if you've discovered something that's new, you don't really have something to compare it to.
Yeah, exactly. And then there's also the kind of bias of almost all work that's been done in this field is in mammals, and a lot of it's been done in humans specifically. So a lot of the way it works is it'll just find the closest match. So if everything in the library has been found in humans, the closest match might be something that's not actually that related to it. We have a couple different approaches of trying to figure out what those bacteria are, their true identity, and then also trying to get at what functions they might be doing for the tuatara as well.
Are there certain categories that the microbes will fall under for function within a tuatara stomach?
Yeah, so I haven't been able to do, this is why I was so excited about the fecal sample, because my cloacal swabs don't have a ton of DNA on them, so I haven't been able to do any functional work from my samples. But the ways around that would be if we had a fecal sample, that's a lot more material that we could work with. But as well, one of my colleagues, Tabitha Caldwell, who works in the lab with me, she's been culturing things from the tuatara gut, so growing them in pure culture. So from that, we'll be able to get a much better assessment of what they are actually doing, because we can get longer chunks of DNA out of them.
And when we talk about function, I mean, microbes in our gut, we know, help us digest our food. Are we thinking it's similar things? And are there also different categories, like things that might not be good for the tuatara? What else might you find in there?
Yeah, that's a good question. So one of the main things I found in the reptile gut more broadly was Bacteroides, which is a genus of bacteria that we also have as well. But yeah, so Bacteroides in the reptile gut and potentially also in our gut can make up something like 1 in 4 bacteria. So it's a very common genus. And the main thing that it's known for is carbohydrate degradation. So big sugars that we eat, that we need for our essential fuel, are very complicated to break down. So they can help break down big molecules into smaller, more digestible chunks for us. And Bacteroides was one of the things that I specifically found almost none of in tuatara. So whatever they do have, I mean, they're not eating that different stuff from other reptiles. So whatever bacteria are in their gut, I'm imagining that either the tuatara doesn't need that function served, or there's another bacteria doing it for them. So that's something I'm pretty interested in finding out, is if the bacteria present are filling the same functional roles that we'd expect other bacteria to be filling that are absent, or if they kind of have their own thing going on.
Yeah, that's totally fascinating. And it's really interesting to think that they might be this little, I don't know, spaceship from a million years old that is the only place that these bacteria can now live, right? Is inside the tuatara stomach.
Potentially, yeah, which is very interesting to consider when you think about how, so all the tuatara that I study are either from Takaporewa or descended from tuatara from Takaporewa, but they haven't been there in like 10 years. And like I mentioned, the gut can change pretty quickly. But what's also interesting is that all of the sanctuary sites, not just the adults, but their offspring as well, tend to show pretty similar bacteria. So if these bacteria can't live outside the tuatara, then how are they being transmitted from parent to offspring? Because reptiles don't have the same level of maternal care that mammals do, and in tuatara it's absolutely none. So they lay the egg, bury it, and then are gone. So the mechanism of transmission there is also something I'm very interested in and have no answer for.
But back to your results, when you say the difference between the wild and captive microbiomes, you know, practically, is that kind of a good result that you see, okay, there's a difference in maybe the volumes of the microbes, but in general, the, you know, spread of what's there is the same. Is that a positive thing that, you know, these tuatara in the ecosanctuaries, you know, generally their microbiome is conserved?
I would say so. Without really knowing what we expect to see, I would say it's a pretty good sign that we see the same thing consistently. Because what that demonstrates to me is that if those are filling an important role on Takaporewa, where the tuatara are from, they're still being conserved when they've been translocated hundreds of kilometers away to an eco-sanctuary or into captivity in a zoo. So to me, that's a really good sign. Oftentimes we would look for is what we expect to see there, but for a species like this where we don't have any expectation, I would say seeing consistent findings across sites is really promising for the health of tuatara.
While overall the gut microbiome looked reasonably similar between the tuatara that were coming from these different places, Cam did see those slight statistically significant differences, and she wanted to explore that a bit more. And there could be different reasons for this. One could be temperature. She's comparing tuatara living near Dunedin to those living in the Waikato and Auckland. But it could also be due to diet. The tuatara are living in areas with different vegetation and are eating different things depending on what their living arrangements are.
when you go into captivity as an animal, your dietary range tends to shrink if you're an omnivore. So the keepers give the tuatara here a wide range of food items, but it's still potentially less than they're eating in the wild, especially tuatara in the wild are going to be eating like skinks and bird eggs and things that these ones are not going to be allowed to eat. So things of that nature. So I was really interested in when you shift the diversity of food items being provided, does that shift the gut?
To test this, she honed in on the tuatara at Auckland and Wellington zoos and at Victoria University of Wellington and put them on a really boring diet.
So we gave them only crickets for 4 weeks to see if just eating a single dietary item produced a shift in the gut microbiome, and then particularly if it shifted the diversity of bacteria in the gut. So my kind of hypothesis was if you're enriching one group of bacteria, potentially that would be at the exclusion of others. But that's not really what I saw, so didn't really see that strong of a shift. Again, P less than 0.05, but microscopic effect size. So no strong shifts were produced by changing the diet.
Which is unexpected. She did this study across the spring and moving into summer. And the surprising thing that she did see was that as that seasonal change happened, after they had gone back to their regular diet, the tuatara microbiome of tuatara in different places around the country seemed to sync up with the same bacterial community makeup. So it seems the main influence might be the seasons, not diet.
And that's what I'm starting to see at Zealandia as well. So it seems like the tuatara gut might start to converge again in their more active season into a set of expected bacteria. So more work needs to be done on that, especially when I get my next round of Zealandia samples back. I'll be looking at that in particular. But there's a lot less inter-individual variation in tuatara than we might expect to see, like, for instance, between you and me. Two tuatara that live in Wellington and Auckland might share more bacteria in common than you and I do, for instance.
The Zealandia study she's referring to is the seasonal investigations she's doing now. In Wellington's fenced ecosanctuary Zealandia, the tuatara are free to roam within a fenced area. While she's got the shift to summer yet to look at, so far she's taken swabs across the autumn and spring to see if there was a seasonal shift due to the change in the tuatara's activity levels. Because while they tend to live in the slow lane anyway, in winter, that's dialed up even further.
Tuatara go into something that's called torpor in the winter, so they just slow down all the way because they need external heat to hunt and then also to digest. So if it's not warm enough, they just kind of won't eat until it warms up enough that they can start eating again. So I don't mess with them in the winter, let them mind their own business. But then come spring, I come back and have a look, because when you think about the mammalian gut, we're feeding our gut pretty much constantly. Like, we only go a couple hours without eating at the most of us. So we're giving our gut a pretty constant flow of nutrients. So in an animal that does basically hibernate, what happens to the gut during that absence of nutrients? So there's been some work done in alligators, for instance, which also hibernate. And it does seem like both the type of bacteria and their functions change during hibernation, which is pretty much what I'd imagine. But interestingly, I have seen some shifts, but it hasn't been anything huge. So there hasn't been a huge shift of now something's here that wasn't here before, or vice versa.
For somebody like you who's into gut microbiomes, this is all very interesting.
Oh, yes.
And you're obviously super passionate about it. But taking a step back, what does this help us understand? And how can we use this practically?
Yeah, so kind of my lab in general, we work on the gut microbiome of threatened species. So a lot of work in our lab has been done on takahe or kakapo, or these animals that have to be very intensively managed. So the idea was, ideally, at some point, tuatara will be able to live all throughout New Zealand again without our intervention. But right now, they can't persist outside of the spaces that we create or maintain for them. So understanding what we expect to see in their gut was really important. So if we looked at the ones on Aotearoa, where they have little to no human interaction, and then look at the ones at Auckland Zoo, where they have lots of human interaction, if we did see a difference there, that would be concerning. But that we don't see one is actually it's reassuring that our current conservation practices aren't interfering with the tuatara gut too much, which is good because we don't really have an alternative right now to captivity and putting them in these sanctuaries. It's kind of the only way to preserve these populations. But the other thing that it tells us with temperature being important is we are already concerned about climate change with tuatara as ectotherms, so they do temperature-determined sex determination.
What this means is that the sex of the tuatara is not sorted by time of fertilization, but instead is determined by the temperature of the soil the egg is buried in about a third of the way through embryonic development. Cool soil temperatures below 21-22 degrees produce females. Temperatures above this result in males.
Additionally, we know that reptiles are really vulnerable to temperature rises because they can't control their internal temperature. Basically, how hot it is outside is how hot they are. They don't have a way to sweat or maintain their body temperature. So understanding if their gut is also affected by temperature, that would mean that they could be more vulnerable to parasitism, they could be more likely to get sick, they could be less reproductively active, things of that nature. So understanding the role that temperature plays in the tuatara gut is pretty important to their conservation moving forward.
And is that next step?
For someone, not for me.
You're in third year,
Yes,
you're wrapping it up, you're putting a nice bow on it.
Yes, exactly, yep.
While she's keen to get her PhD done and dusted, Cam would like to stay in research. So it's likely you've not heard the last of her in this space.
Doing host microbe work is super fascinating for its ecology and evolution, but whenever it can intersect with conservation like this is really important to me. So love to stay in this field moving forward, and New Zealand's a great place to be doing this research.
Thanks to Chye-Mei Huang of Auckland Zoo and Cam Hoffbeck of Waipapa, the University of Auckland. This episode was produced by me with help from Ellen Rikers and Liz Garton. Sound engineering was by Steve Burge, and it was recorded with the help of Otago Access Radio. Photos and more information can be found in the web write-up at rnz.co.nz/OCW. If you've got feedback or episode suggestions, you can send them to ourchangingworlds@rnz.co.nz. Te nā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, te wiki.